Hi everybody, let's continue our discussion of growth in batch cultures. So as we discussed in the last lecture, that primarily used models set up for doing growth experiments in bacteria and other single cellular organisms is batch culture. So let's take a look at a particular growth curve a little more deeply. So again, our setup is going to be that we have a flask. This has a certain volume and we seed this flask with a bacteria, and this is shaking and temperature is 37 degrees Celsius, and presumably there are nutrients here that as we move forward in time, the number of bacteria is going to go up, the number of resources available in the flask are going to go down. That's intuitive. We'll try to give this a little more mathematical sense in this video and the next one. So our goal here is today is to understand sort of some of the kinetic features of this growth process. That what's going to happen as time moves forward, the number of bacteria in this flask. And this is growth curve. This is referred to as growth curve of a microbial species. And this characteristically has a few different phases associated with it. And let's just draw this and then we'll see. So as you can see that there are roughly, this can be divided into 4 different phases. The first phase is where really no growth takes place. So let's say this is till time t naught. And the characteristic feature here is that no to very little growth. In the next phase, let's say up until this point, let me redraw this a little bit accurately. Something like this. In this next phase, growth is happening exponentially. So this is called the exponential phase of growth. In the third phase, let's say this happens till time t1. In the third phase, t1 to t2, the numbers of bacteria in the flask more or less stay constant. So this is the phase when all the nutrients have been used up. So this is number of bacteria stay more or less constant. And in the fourth phase, there is active death taking place, and as a result, number of bacteria decreases with time. And if we let this go on for a sufficiently long amount of time, we'll reach a stage where there are no viable bacteria left in the flask. Let's try and understand what is happening in each one of these phases. So what we discussed in the last video was that the carbon source that's present here is lactose. Lactose is a disaccharide of glucose and galactose. So we have, let's say, the circle represents glucose, the square represents galactose. So when glucose and galactose are linked together, the resulting disaccharide, which is 2 sugar molecules is referred to as lactose. And the way E. coli utilizes lactose for growth is, first it has to internalize it. So, let us say this is an E. coli cell. First it has to bring the lactose inside the cell. So, this transport process has to take place. That is step number 1. And the second step that has to take place is that this lactose has to be broken into glucose and galactose. So, the bond that links the 2 sugars has to be broken, and now the cell has access to free glucose and free galactose, which will then be utilized by the cell for growth and energy purposes. E. coli and most other species cannot use, well, no other species can use lactose as it is. It has to be broken down into glucose and galactose. And that is step number, this breaking down of lactose into glucose and galactose is step number 2. But these 2 steps are facilitated by specific proteins in E. coli, and the first step is facilitated by a dedicated transporter whose name is LAC-Y, and this protein, its only job is to transport lactose from outside the cell to inside the cell. And this protein, as we saw in one of the previous lectures, is only produced when lactose is around in the environment. If you place E. coli in an environment where glucose is the only carbon source, then LAC-Y will not be made in the environment. This hydrolysis of lactose into glucose and galactose is facilitated by another protein called LAC-Z, which also is only synthesized when cells are in an environment where lactose is present as the carbon source. When we first seed this culture with this one bacterium, let us imagine that this is coming from, so this bacterium was taken from a condition where it was growing on glucose, and then we placed it into a flask where the source of energy and carbon was lactose. What that means is that when I seeded this culture, there was no LAC-Y or LAC-Z in this cell that I placed in the flask. As a result, because it lacks the necessary proteins to grow on lactose, there is going to be no growth in the culture that I witness, because the cells that I placed there are not ready to utilize lactose, because they do not have these necessary proteins for breakdown and internalization of this disaccharide. This not being ready to utilize the resources that are present in the current environment leads to this phase of no growth, and this is referred to as the lag phase. That is the first phase of growth that characterizes bacterial growth in a batch culture, which is lag phase. After a while though, the cells, in practical terms, seeding will not happen with one cell, there will be several cells that I seed with, and hence after a while these cells would have made sufficient amount of LAC-Y and LAC-Z to sort of start exhibiting growth. And in this condition where initial density is low, so at this point when cells have made LAC-Y and LAC-Z, number of cells in the culture are small, nutrients are plenty, and as a result they start dividing exponentially. So, 2 become 4, 4 become 8, 8 to 16, and so on and so forth. As a result, with time this phase of growth exhibits this exponential increase in number. Hence, for this reason this phase is called either the exponential phase or the log phase of growth. And this is the second phase associated with bacterial growth. But soon what is going to happen is that because the number of bacteria in this flask are growing exponentially, and with that exponential growth every individual cell is placing its demand on lactose and other resources that are available in the flask, resources are going to become very limited, and as a result this growth will start to decline, and after a while there is no more growth taking place. and as a result the number of bacteria in the flask is more or less constant, and this is why this phase of growth is referred to as stationary phase of growth. And in the stationary phase of growth, what happens here is that number of births is exactly balanced by the number of deaths taking place. So, what is happening in stationary phases is not as if there were n number of individuals and they n stay the same throughout, but what is happening is that some bacteria die. When they die, they lyse open, the constituents of these bacteria are released into the extracellular media, and these constituents provide some resource to facilitate a minuscule amount of growth. So, in this phase of growth, the births that are facilitated by death and lysing up of cells which are no longer alive, these two processes are balanced with each other and leading to the overall number of bacteria in the flask staying constant, and this is referred to as stationary phase of growth. But this is not sustainable. Eventually the stress of not being able to grow and maintaining homeostasis is going to be too much, and numbers are going to decrease in this death phase as we move forward in time. And depending on how deep we go here, this is going to eventually go down to 0, where there will be no viable bacteria left in the flask. So, what we should focus here is that in these 4 phases of growth, let's say this is phase 1, phase 2, phase 3 and phase 4. Bacterial growth is characterized by its performance in each one of these 4 phases. So, we will try and understand some numerical estimates of what allows us to define this growth process. So, again let us go back to our growth curve and we get something like this. So, in the first phase, this is lag phase. The characteristic of lag phase is the duration that the bacteria needs for it to start exhibiting growth in the environmental condition that it is being thrown into. So, this is characterized by its duration. So, the relevant parameter here, because lag phase by definition does not exhibit any growth. So, what defines lag phase is the amount of time it takes bacteria to break out of this phase where there is no growth and start dividing. So, it is characterized by a duration and hence it could be measured in minutes or hours. Some unit of time. What you should note at this point is that suppose I have a flask and this flask contains lactose as the carbon source. In that case, when I seed this with one bacteria, if this one bacteria is coming from an environment where it was growing on lactose, in that case, because in the previous flask from wherever it is coming, because it was growing on lactose, it already had lac Y already present in it and also lac Z. So, these two were present in it, and hence when I transition it to another flask which also contains lactose as the carbon source, lag is going to be very short. Because it was already going in an environment and I am simply placing it in an environment with the same amount of resources, with the same resources, only its quantum is different, and hence it does not need any time to make these proteins and then start utilizing lactose. It is already ready to use lactose and exhibit growth. On the other hand, if this individual came from an environment where it was growing on glucose, this individual does not have any LAC-Y or LAC-Z, and hence it is going to need some time to make those proteins, and only then it will be able to exhibit growth. So in this case, lag is going to be long. This could be several hours before it starts to exhibit growth. But what we should take into account here is that this duration of lag is independent of the genotype. Their DNA is exactly the same, but the duration of the lag is different in the two cases because of the environment from which they are coming, not because of the genotype that they are carrying. The genotype of the two individuals is exactly the same. So, lag phase duration is measured in time and is dependent on the prior culture conditions. That is not to say that it is completely independent of its genotype. It is obviously also dependent on its genotype. For instance, if these two genotypes were different from each other and this purple individual carried a mutation such that LAC-Z is no longer working in this. If LAC-Z is no longer working in it, that means this individual can never make LAC-Z, which means that hydrolysis of glucose and galactose into hydrolysis of lactose into glucose and galactose cannot take place in this individual, as a result of which this individual can never exhibit growth in an environment where lactose is the only carbon source. So, and that results because of this mutation that it is carrying and hence its genotype. So, duration of lag phase is dependent on prior culture condition and the genotype of the organism, and is measured in amount of time it takes an individual genotype to start exhibiting growth. Let us move to the next phase. In the exponential phase or the log phase, growth is exponential, and as we will see, let us say that the number of bacteria at the beginning of exponential phase is n naught. Then growth is taking place exponentially, and hence in this phase the number of bacteria at any given point in time, let us say Nt, is simply equal to n naught into e to the power RT. What that means, let's write this a little more clearly. In the exponential phase, Nt is equal to n naught e to the power RT, where Nt is the number of bacteria at time t. So, this is in context with our growth curve, and we are talking of this particular phase, and this is the number of bacteria at the beginning of the exponential phase or the log phase. And let us say this, as far as the log phase is concerned, this time here is t equal to 0, and at any given point time t in the log phase, if we were to estimate that how many bacteria are here, that is Nt, this number can be estimated using this formula, where Nt is the number of bacteria at time t since start of log phase, N naught is the number of bacteria at beginning of log phase and we have one more variable here which is R, which is the growth rate, which means that once the bacteria starts to divide in the log phase, how fast does it divide? So, faster growing variants, faster the growth, greater the R. Because as far as log phase is concerned, we have one bacteria which divides into two, but then this divides again and we have 4 and so on and so forth. This is the definition of log phase. However, what log phase does not tell you is that how long did each one of these processes take, T naught. If this is happening really rapidly, if growth is happening every 20 minutes, then that's a very high value of R. However, if it's taking 3 hours, then that's a smaller value of R. So, in this log phase, growth can be estimated via this formula. All right. In the next phase of growth, so first phase lag is characterized by its duration, log it's characterized by R, its growth rate, stationary phase is also going to be characterized by its duration, which is to say that once the bacterial population has reached a certain density, once this has reached a certain density, let us say after T dash amount of time, how long is it before numbers actually start falling? Let us say this is T double dash. So, what is this duration of time delta T before numbers start decreasing? And what you should realize here is that in this stationary phase, the death process is actually exactly balanced by the birth process. However, beyond stationary phase, the number of deaths is greater than number of births. So, what we are asking here is that how long does the population carry its resilience such that the death and the birth processes are closely matched with each other, beyond which death rate is going to exceed the rate at which births are taking place and overall number of bacteria are going to decrease with time. The longer the stationary phase associated with a species, it is in some sense a representative of how resilient is the species to tackle the stress and not let death rate exceed the birth rate associated with the process. So, this is sort of these 3 important phases of growth that we will be focusing on. Let us now do this toy example where let us imagine that we have these 2 species. We are going to represent time against number of bacteria. So, species 1 exhibits a growth curve like this. that if we start the species 1 with n naught number of individuals, it exhibits a certain lag phase duration and then it exhibits exponential growth, but then there is stationary phase followed by death at a certain rate. So, that is the characteristic feature of growth associated with species 1. Let us compare this with species 2, which is also seeded at the same number n naught, and let us say that species 2 exhibits a slightly different growth characteristic. And so on and so forth. This is species 2. So, now the question before us is that if individually grown in a flask, if these two species are individually grown in a flask, that is the characteristic feature of growth that they exhibit by themselves. But if I were to collectively grow them together, so let's pose this question as the following, that which of the two species is fitter if grown together in a flask. And as we can see that the answer to this question is not as straightforward as it might appear. If we compare their characteristics in the growth phases, so we have lag, log, stationary and death phase. We have species 1 and we have species 2. If I am looking at only the lag phase, then up until this point there is no difference between the performance of these two species. However, if I am looking at the end of log phase of species 1, then I note that the log phase associated with species 1 is longer compared to the log phase associated with species 2. So, by the time species 1 starts to grow, species 2 has already started growing for some time, and as a result, if the comparison that I am making for fitness is at the end is during the log phase, then 2 is fitter than species 1. So, if the comparison is in lag phase, species 2 is fitter. And again I will repeat the reason, because in this lag phase species 1 is not growing at all, whereas species 2 starts to grow faster earlier as compared to species 1. So, while species 1 numbers remain constant at n naught, species 2 numbers start to increase before species 1 enters its log phase. Secondly, now if we move to log phase, we note that species 2 grows at a rate which is much more rapid as compared to the growth rate which is exhibited by species 2. So, 1 is growing faster as compared to 2, as a result of which if my comparison of fitness now happens in a log phase, then species 1 is winning. Next, we said that stationary phase is a measure of some sort of a measure of resilience of a species to keep the death rate balanced exactly by the birth rate. If we look at how long these stationary phases are for these two species, for species 1, this is the duration of the stationary phase. Of course, because it was growing very rapidly, it entered stationary phase faster as compared to species 2, whereas for species 2, the stationary phase is much longer as compared to that of stationary phase. So, let us say this is delta t for species 1 and this is delta t for species 2. Delta t2 is greater than delta t1. So, as far as stationary phase is concerned, species 2 is able to keep its number constant for a much longer time as compared to species 1, because after delta t1 time species 1 numbers start to decrease. So, if we are comparing fitness in stationary phase, then species 2 does better as compared to species 1. And lastly, when they enter death phase, we see that the fall in numbers for species 2 is extremely rapid. So, while it maintains its stationary phase for a long time, its fall in numbers is extremely rapid and it goes to 0 in a very short amount of time. Compare that with species 1, where the numbers are decreasing much more gradually, and hence it's much more, it's able to resist death much more as compared to species 1. And as a result, in death phase, species 1 is fitter as compared to species 2. So, the point of this exercise being that now we have these 4 measures of growth and fitness in a simple growth curve, and when we compare these two species, sometimes one will do better as compared to the other in some performance criteria, whereas in another performance criteria other species will do better. So, it's not clear looking at this table that which is fitter. Because although if I do the growth curve till here, although species 1 is fitter, but in the preceding 3 regimes that were there, species 2 was doing better. So, what is going to be the sum total of fitness effect of all of these 4 phases of growth that I am looking at? For this reason, we need to develop some quantitative understanding of how to compare growth curves and what would happen if I grew them together in a single flask, and that is what we will continue with in the next video.